So now in last class we discussed about different varieties of transport mechanisms. We discussed active and passive transport. Now today we'll discuss about special categories of transport, which includes bulk flow, vesicular transport, which is further divided in endocytosis, exocytosis and transcytosis. Then filtration and osmosis. Now let us start with first one, bulk flow. It is passive transport, it is diffusion. Here substances are transported from higher concentration to lower concentration only. But here the name itself suggests bulk because they are in large amount, that is bulk flow. For example, gases, respiratory gases are transported by this mechanism. Second is vesicular transport. Here transport across the cell membrane by formation of vesicle. A vesicle is formed and this vesicle is being transported. Here energy is required from ATP. This again we have discussed, it is of three types: endocytosis, exocytosis and transcytosis. Starting with endocytosis. Endo means entry inside the cell. So macromolecules, they enter inside the cell. Here there is infolding of the membrane. Membrane is folded inside and formation of vesicle. Now it is again of three types: pinocytosis, phagocytosis and receptor mediated endocytosis. What happens here in pinocytosis? This is also known as cell drinking because molecule is smaller than phagocytosis. So here, but the molecule is macromolecule like bacteria or other antigen. And it is also seen in some specific cells like digestive tract, in kidney cells. Now, how this happens? This macromolecule you can see here, first of all it binds with the outer surface of the cell membrane. Okay, and there is depression, or you can say there is evagination of the cell membrane, and the droplet is engulfed by the membrane. This engulfed droplet that is converted into vesicle. You can see here this is a vesicle, or it is also known as vacuole. And this vacuole or vesicle, this is known as endosome. Now after that what happens? Primary lysosome binds with this endosome and it causes digestion and degradation of the particle of the endosome. So this is the mechanism of pinocytosis. It is a type of endocytosis. Second one, that is phagocytosis, which is also known as cell eating. Here the molecule is larger than the molecule for pinocytosis. Here what happens when molecule that is larger than macromolecule like bacteria, dead tissue or foreign particle, when they are there in contact with the cell and we require to make them enter inside the cell. This is by the mechanism of phagocytosis. Only all the cells, they don't have phagocytic mechanism. Only few cells like neutrophils and macrophages, they have phagocytic property. Now what is the mechanism? Here you can see this is phagocytic cell. First of all, this phagocytic cell, it gives pseudopodia. You can see false projection like this. Pseudopodia, yeah, or they are cytoplasmic extension. They are produced. And they again cover the particle, and these particles, they are engulfed by the cell, and again there is formation of endosome. This endosome is known as phagosome, and again fusion of primary lysosome with the endosome, and this endosome is degraded. Third variety is receptor mediated endocytosis. Here what happens? You can see the cell has got clathrine coated pits. This is a pit which is coated by the clathrine. Okay, and they have receptor proteins here. You can see. Now, when the substance, which is also known as ligand or hormone, when it binds with this protein, this is a protein which is present in the pit, and this pit is coated by clathrine. So, clathrine coated pit has receptor. Okay. Now what happens when ligand or the hormone, it binds with this receptors? Okay. Now there is, after binding, again this pit is detached from the cell membrane and the whole receptor along with your ligand and the substance. What happens next? That is, pit is detached from the cell membrane and again there is formation of vesicle. This vesicle again is known as endosome, and this endosome enters inside the cell. Here only difference is we have this receptors. Okay. Again lysosomal enzymes, they digest this endosome. So this is receptor mediated endocytosis. Next is exocytosis. Here exocytosis, in which the substance moves outside the cell through the cell membrane. This is reverse of endocytosis. Endocytosis means entry, exocytosis means exit, and it is present in the secretory cells. Secretory cells, they have secretory substances which will be excreted out. Now, what is the mechanism here? The vesicle containing the secretory substance, first of all you can see this is a cell. This is our vesicle. It comes in contact with the membrane. Now fusion of both the membrane of vesicle and the cell membrane. Now this gets dissolved and the substance, either it is hormone or it is enzyme, that is excreted out. Now it is by two pathways. Number one, that is non-constitutive pathway. What is it? In this pathway, the substance or the protein which is secreted from the Golgi apparatus, initially it enters in the secretory granule and there is processing. So non-constitutive, here what happens? Processing takes place. Processing takes place. So that is another name of this non-constitutive pathway is, it is known as regulated pathway. Example is secretion of hormones, neurotransmitters, some digestive enzymes, they all are secreted by this pathway. Other is constitutive pathway. In constitutive pathway, there is no processing. Here the proteins are transported directly from vesicle to the exterior. So this pathway is known as non-regulated. This may be asked as your MCQ, regulated and non-regulated pathway. Regulated is non-constitutive, non-regulated is constitutive. So this is exocytosis. Next is transcytosis. Here what happens? Suppose this is a cell. So endocytosis from this side and exocytosis from other side. Sometimes receptor mediated endocytosis may be there. So the substances, they enter from one side and exit from other side. Other names from transcytosis is also known as cytopemphasis. Cytopemphasis or vesicle trafficking. Okay. Example is, for example, some substance is there in the blood. It enters in the endothelial cells from one side and it exits from other side. So this is transfer of substances by transcytosis. Next is filtration. Here what happens? Filtration means fluid that gets filtered. So water or fluid, it moves from high hydrostatic pressure to low hydrostatic pressure. Hydrostatic means pressure of the water. Suppose here we have a large amount of fluid. So water head pressure, that causes filtration. Example, fluid is filtered from blood to the interstitial space, or in the kidney. We all know there is Bowman's capsule. Here there is glomerulus. So filtration takes place here. Next variety of transport is osmosis. This is also diffusion, but this is special type of diffusion. What happens? What is the difference between diffusion and osmosis? Is here in osmosis there is semi-permeable membrane between two substances. Here in osmosis there is presence of semi-permeable membrane between two liquids. You can see here like this. Okay, and the membrane is semi-permeable means it is permeable to fluid, not the solute. So it is permeable to solvent, not the solute. So here what happens in osmosis? There is passive movement of solvent. Solvent moves from low solute to high solute concentration. Low to high solute concentration. Yeah. Here important requirement is solute concentration must be different on both the sides. Now endosmosis and exosmosis, these are two terminologies. Endosmosis means entry of fluid inside the cell, entry, and exosmosis means exit of fluid from the cell. Now we have osmotically active particles. This terminology is used for the solutes. They cannot pass through the semi-permeable membrane and they create pressure, and the pressure created by these particles, osmotically active particles or solute, is known as osmotic pressure. Now this osmotic pressure depends on the number of particles, not on the size, not on the type, not on the composition. Okay, so if numbers are more pressure is created. One more terminology here is osmole. What is osmole? That is molecular weight of substance in gram divided by number of freely moving particles. For example, if we have a solution of molar solution of sodium chloride, so it has got sodium and chloride, so it is 2 osmole. Okay, one is sodium and one is chloride. Now what is osmolarity and osmolarity? That is also important terminology. Osmolarity means number of osmotically active particles per liter. Osmotically active particles per liter, that is osmolarity. And osmolality means number of osmotically active particles per kilogram. But normally we use the terminology liter, so we use osmolarity. Now other terminologies are isotonic fluid, means osmolarity same as plasma, same as plasma. What is the osmolarity of plasma? That is 270 to 290 milliosmoles per liter. Then what is hypotonic? When the osmolarity is less than plasma, less than plasma. When it is greater than plasma, that is hypotonic. Now what is the importance of isotonic solution? Isotonic means same as osmolarity, same as plasma. So if our RBCs are placed in isotonic solution, they are of normal morphology. They will not shrink or not swell. If we are putting the RBC in hypotonic solution, means suppose you can see these are the RBCs, or we are injecting hypotonic solution. So what happens? Fluid moves, osmosis of the fluid inside the RBC. So RBCs, they got swollen, okay, and they rupture. And if we are putting our RBCs in hypertonic, what happens? Because here solutes are more, here solutes are less, so osmosis of fluid from inside to outside. So RBCs, they get shrink. Now one applied aspect here is if somebody has got severe dehydration, what happens? Water is lost, so osmolarity increases. In patient of dehydration, osmolarity increases. In patient of diabetes mellitus also osmolarity increases. If the person is having hyperosmolar coma, means osmolarity is increased. When osmolarity is increased, what happens? What happens in hyperosmolar coma? Hyperosmolar means osmolarity increases. So from brain cells, fluid comes out and the patient will be in coma. If the urea level increases, for example, like person is having renal failure, then also plasma osmolarity is increased. And hypertonic solution mannitol, that is mannitol is given sometimes in case of cerebral edema. And what is its importance? It causes osmosis of water or fluid from interstitial space to the blood, so to reduce edema. Then one more terminology is colloid osmotic pressure. Osmotic pressure we have discussed. Osmotic pressure, that is pressure exerted by osmotically active particle. But colloid means they are plasma protein. So plasma proteins, they exert osmotic pressure in our blood or plasma, that is colloid osmotic pressure. Okay. The flow of water inside or outside the capillary depends on the colloid osmotic pressure. If the pressure decreases, then the fluid will move fast. And normal colloid osmotic pressure is about 25 to 30 millimeter of mercury. So colloid osmotic pressure is important to prevent edema. Okay. Then another is solvent drag. Solvent drag means there is movement of solvent into or out of the capillary, which carries dissolved particles with it, and the force, it is known as solvent drag. So this is all about our various transport mechanisms. Thank you.
If you wish to get all topic notes, my book Physiology Notes is available on Flipkart as well as Amazon, and link for both that is given in the description box. And to get the soft copy, my application Vesalius, that is available on Play Store. You can download it and get the subscription. Link for that is also given in the description box. Thank you so much.